And what I did is I took a couple of slides, I think I took most of the latter half of the other lecture, and I put them to this lecture because one is I want to repeat it, and the second one is I just want you to understand what's going on here. So let's go back one step, and the step is cross-bridge cycling. Okay? You're going to have this myosin head sticking out of the myosin. It's going to come in close contact with an actin. Okay? The minute you get calcium, that releases the inhibition on the tropomyosin, and then the myosin head attaches. Before the myosin head attaches, it already has the energy it needs to bring about the contraction. Okay, and I keep using this word contraction. What it has is the energy to bring about the power stroke. In other words, the pulling. Okay, which if all of them pull kind of together, then that pulling will cause the contraction. So it binds, calcium, it binds, and the ADP and the PI is kicked off the myosin head, and now you got the power stroke. And now you need the ATP. The ATP comes in, detaches it, and you can go through the whole cycle again. Essentially what you're doing is you're bringing the sarcomere closer and closer and closer to each other. Sorry, the Z line closer and closer, you are shortening the sarcomere. And if all the sarcomere is shortened, you shorten the entire muscle. Now we say the muscle is contracting or shortening. Okay? Good. And so that's what I'm going to look at. And again, if you look at that in terms of, okay, I need an action potential. So the yellow line is the motor neuron. It comes to the end plate. You then got the acetylcholine. You've got the action potential in the muscle. That now is spreading, and that's shown by the black line. The effect of that thing spreading will give you the action potential in the muscle. So the action potential in the nerve, then you got the action potential in the muscle. As it's going through the muscle, you're getting this thing between the DHP channel and the ryanodine channel, and you're getting calcium release. And now you're starting the cross-bridge cycle. And there's the contraction. In other words, the cross bridges are pulling and then shortening up the sarcomeres, eventually the entire muscle. There's the contraction. Now the calcium is being pumped back into the sarcoplasmic reticulum, and then you get relaxation. This is called one muscle twitch, one muscle twitch. One contraction, one relaxation of that group of muscle is called a muscle twitch. Again, you got this latent period simply because you need to accumulate sufficient calcium in the muscle so that you can bring about the cross-bridging thing. I then went on to say, okay, there are three important things that affect the tension that might be formed in a muscle. And the first important thing is the number of cross bridges that you could potentially get between the myosin and the actin. So you've got the myosin, you've got the actin. So if I have three possible areas I can attach, that's the amount of tension I can generate in that one. The second thing is, what is the resting length of the muscle and therefore the sarcomeres in the muscle? Okay, that's the second thing that will affect how much of tension I'd be able to generate in the muscle. And the third thing which I look at today is this action potential coming down the motor neuron, the red arrow. So let's start with the first one. Okay, so I mentioned to you that the cross-bridge cycling, so you got the myosin, you got the actin, it holds, it pulls. Again, if I have lots of possibilities of making cross bridges, that means I'm going to get a decent amount of tension. Just makes common sense, yes?
Yes.
So if I look at that cross-bridging thing in terms of sarcomeres, these individual units that are in the myofibril, this is what I will get. So, look at this graph, busy diagram. What this thing is saying, if you look at the y-axis, it's the amount of tension. If you look at the x-axis, it's the length of the sarcomere. And what it's saying is that at this particular length of the sarcomere, I will get the greatest amount of tension in that sarcomere. Are we okay?
Yes.
Good. What is particular about this length of the sarcomere? Firstly, what are the numbers here? It's approximately 1.98 or 2 to about 2.2 micrometers. That's what that is. Are we okay? Good. Now, why that particular length? Because if you look at that particular length, now you go to the top diagram. You notice that the possibility of forming cross bridges, because you know the blue line touching the red line? Yes? Can you see that?
Yes.
It's showing you how many times you can possibly meet each other. It's already attached. So if you're looking at this one, you got 1, 2, 3, 4, 5, 6. Okay? The possibility of attaching. Are we okay?
Yes.
So that's the number of possibilities you have for the myosin and the actin to attach to each other. Obviously, the greater the number of possibilities for attachment, the greater the force. And that's what it's telling you. At this length, you will have that amount of force. Now, let's stretch the muscle. Let's stretch the muscle. So I now come here. Okay? So I'm stretching the muscle, the sarcomere. When I stretch the sarcomere, notice the possibility of forming cross bridges has decreased by half. There I had how many? 6?
6.
Now it's decreased by half. That means the amount of force I will generate would be less. If I stretch it even more, I can't get cross bridges. It's too far stretched. So it's here. On the other hand, if I squinch it, in other words, I push it in, okay? Now what starts to happen? Look at it again, look. The possibility of getting cross bridges starts decreasing because if you look at, say, this one, see it's coming through and it's interfering. This actin is interfering with that actin. So now I'm getting less force again. So at the level of the sarcomere, individual sarcomeres, it's telling you what level of force a muscle can generate, or at least a myofibril can generate. And that's what this diagram is saying. So if you keep the muscle at that optimal length, that LO, or a little bit below and a little bit after, at the bottom it says about 75%, which is a little bit below, and about 130%, that will give you your greatest amount of force because you have the greatest amount of overlap, possible overlap between actin and myosin. Does that diagram make sense?
Yes.
So that's at the level of the sarcomere now. Can I extend that same kind of thing on an entire muscle? Yes, I can. So this is for the whole muscle. Now, I'd like you to look at me for a minute, please. So there's my bicep. Can you see my bicep? Stop laughing at my bicep. Okay? So, there's my bicep. My bicep is listing nothing right now. It's relaxed. Yes? And if you look at that bicep, in this relaxed state, we'll call it length 1.0. Are we okay?
Yes.
Are we all right?
Yes.
Now, this is what I'm going to do. My bicep is still relaxed, but my tricep is contracted. It pulled the bicep down. Is the bicep still relaxed?
Yes.
Yes. Yes, I'm not lifting anything on it. Yes?
Yes.
That means I've moved it to position number 2. I have, at least the tricep contracting has stretched the bicep. Are you with me?
Yes.
Now what happened in that motion? What happened going from here to here? Look at the graph now. Okay? So this is where was at the 90-degree angle. When it got stretched, the passive tension started to increase. I'll tell you what that is in a second. Okay? So the passive tension started to increase. When I got to 2, in other words, like this, all right, it's still not contracted, but now the passive tension is high. Okay? Now, so come back, have a look at this bicep. So there's the bicep at position 1.0. Let's say you could stimulate that bicep by putting 2 electrodes on there and giving me a slight shock. Are you with me? Yes. I keep my hand like this, but what will be the force in that bicep? It will be in the black graph. Can you see the black graph?
Yes.
Okay? Are you with me?
Yes.
So it'll be that thing in the black graph. And then you tell me, okay, move your hand like this. You stimulate again. I get the black graph. All right? You then say, do this. You stimulate, and then I'll get the black graph going on the other side. What is that black graph? It is that passive tension plus whatever contraction there is in a muscle. That's that total tension, the black one. Now I need to calculate how much of tension or force was actually created by the actin and the myosin moving over each other. We call that the active tension. How do I do that? I subtract the red graph from the black one. So I subtract this number minus this number, whatever it is. That number minus this number. This number minus this number. You understand what I'm getting at?
Yes.
And what do I now get? I get the dotted line, the active tension. That's the amount of tension or force generated by the actin and the myosin moving over each other. Again, notice it follows the similar kind of pattern as the sarcomere. The active tension follows a similar pattern as the sarcomere. So the entire muscle must also be at a supposed optimum length, which is given in my diagram by this yellow bit in the middle. Okay, in that yellow bit, you will create the maximum amount of tension or force, whatever word you want to use, if you stay in the yellow bit. If you go below the yellow bit, you have less tension. If you go above the yellow bit, you will have less tension as well. Yes?
Yes.
And that's what I wrote for you here. The muscle length must match the maximum tension. That is called the optimum tension. If the muscle is either shorter or longer, I'm referring to the muscle, not the sarcomere. If the muscle is either shorter or longer, you have less tension in the muscle. Now, if the total tension is the sum of passive and active, and if active is the actual muscle contracting, now what is the passive tension? We call it the preload. In other words, it's a stretching of the muscle. Okay? So it's the stretching of the muscle that creates the preload. And again, if I go on to the next slide now, okay, who gives the muscle its passive tension? It is all of the connective tissue, the cartilage, the connective tissue, and so on, around the muscle cells, inside the muscle cells, in the tendon, and so on and so forth. That's what creates that passive tension, if you want. Okay? And it just so happens that that passive tension actually occurs in the opposite direction of the actual muscle contraction. Okay? So, if you had a muscle like this, and it's attached to a bone here, okay, and the muscle is attached to a bone there. The passive tension is in this direction and in this direction. You're pulling it. Are we okay?
Yes.
But the actual contraction between the actin and the myosin is in this direction. It's like that. That's mine? Okay. The actual contraction is in this direction, but the passive tension is actually pulling outwards. But when you add both of them up, you get the total tension. Okay? So, again, the elastic components, because they are in series, you have to overcome them before the actual load is moved when the muscle is contracted. If the load is moved, that means it's an isotonic contraction. If the load is not moved, the muscle is not contracting sufficiently, then it is an isometric contraction. And I try to represent that in this diagram. So if you're looking at this thing now, you've got this muscle, its maximum load it can carry is 4 kg. Okay? I put a 2 kg load on that muscle. So you got the muscle, you got the thing out there, and now you stimulate the muscle. Now, before you stimulate the muscle, the minute you put the 2 kg, it's going to have a passive tension. Are we okay? Good. Now we stimulate it. When you stimulate it, again, the afterload is pulling it this way, but then the contraction is pulling it this way. Are we okay? But that contraction is bigger than the afterload, so this thing moves off the table. Are we okay? Good. So if you look at that in a graph now. So there we are, we put the load on, we stimulate here, the tension slowly increases, it goes to a maximum of 2 because that's what we have. Yes? It stays at 2, the muscle then relaxes and then it comes down. But if you look at the muscle itself, and here you got muscle length as a percentage of the rest. So it was this size, okay? Now as the muscle started to contract, what happened to the muscle? It became shorter and shorter and shorter, all right? And then when you have relaxation, it comes back to normal. Now, because the load moved, we call that an isotonic one, okay? Come to this one now. Because this muscle can only carry 4 kg. Are we okay? We put a 6 kg load on it. So there we are, there's the muscle. We put a 6 kg. It has a high passive tension now. It's stretched. We stimulate it. All right? And it now starts to contract. Can you see it's bulging? It's contracting. All right? And as it's trying to contract, contraction is not sufficient enough to lift this thing off the table. If you look at the graph again, stimulate here, it gets up to 4 because that's the maximum it can go to. It stays at 4. The amount needed to lift the load is actually 6. So it is contracting, but it stays here. All right? And then the muscle relaxes and you come back. But look at the length of the muscle. It remains the same. Okay? The length remains the same. Again, why? Because it's sitting on the table. All right? So you got that isometric contraction on the right-hand side. Okay. Because a muscle is contracting, how fast does it contract? Is the question. And the question now is, okay, if you ask me to lift up this pen and to flex my elbow, right thing, I will do that. Yes? So, if you ask me to lift this, maybe this is a little bit heavier, it'll be a little bit slower. Yes? So, in the sense of using my arm, I'm essentially looking at how fast my forearm comes closer here. Is that okay? Yes. That would be velocity, say. So, if you are looking at velocity, so you've got velocity of contraction, how fast the muscle contracts, against the load or the afterload that you have on it. Notice here, the velocity is 0. Yes? Yes. Because the load is so great that the maximum force your muscle can generate will not move it, and we call that an isometric contraction. Are we okay? Yes. Good. Now come here. Here the muscle is contracting very, very fast. And the reason it's contracting very, very fast is because there's no load on it. Yes? Yes. You know, it's like nothing to pull on. All right? So it's contracting very fast. So when you have no load, the muscle contracts very fast. When you have a load that is so great that the maximum contraction of the muscle cannot move the muscle, that is called an isometric contraction, and F stands for force, the maximum force. And if you look at the graph, notice it's a hyperbolic graph. Okay? So those of you who did math, AP math and calculus math, will notice that there are two straight lines, the one at the bottom and the one here. So when you come here, the muscle changes very quickly. When you are here, the muscle changes very slowly. Okay? You don't need to know the math. All right? The only thing you need to know is when the tension of the load equals the force of contraction of the muscle, then you're going to get zero velocity. On the other hand, okay, when you have no load on the muscle, you'll get extremely fast velocity. Obviously, as the afterload decreases, you're going from the bottom there this way, as the afterload decreases, the velocity of shortening will increase. In other words, it's an inverse relationship. Okay, so I then had this table which I kind of summarized for you. Preload will be the initial resting length of the muscle before it is stimulated to contract. Okay? And again, what does it determine? It determines the maximum potential force that the muscle will be able to generate. Remember the LO, you can get to the highest point at that preload. Okay? Afterload is the load or the weight or the mass, whatever it is, you are trying to carry. What does it affect? It affects how fast the muscle will contract, in other words, the velocity. Okay? The velocity, high afterload, you get a slow velocity. Velocity of contraction, the speed at which the muscle will shorten, again determined by the afterload. Lastly, the amount of force generated by the muscle is determined by the preload, or the stretching of the muscle, or another way of saying that is the number of potential cross bridges you can form. Okay. I now need to go on to number 3 in my little table. And the number 3 was, if you remembered, I had a motor neuron coming to the muscle, yes?
Yes.
What will the effect be if we change the firing pattern on the motor neuron? And that's what I want to look at now. So if you're looking at the diagram, I got an action potential coming down, okay, on the muscle. So I'm stimulating once. It contracts. I then pump all the calcium back in to the sarcoplasmic reticulum. It relaxes. Are we okay?
Yes.
Notice there is no any more contraction here to actually stop the calcium from being pumped back in. I'll tell you what I mean in a minute. So now I come to the next bit. I give a stimulation, there's my contraction. As it's coming down to relax, I give another contraction or another action potential test. Are we okay? What happens now? It goes up from there. In other words, the relaxation doesn't go all the way down and start all over again. Yes? Why is this happening? Because this first contraction released a certain amount of calcium. Are you with me? Now that calcium is being pumped back into the sarcoplasmic reticulum to cause the relaxation. It's coming down, down, down. Then you get another action potential. What happens to the calcium? It bumps back up like this. Therefore, you get this thing here. Now, if you bring these two very close to each other, they actually fuse up, so you cannot see this and that as separate, you just see them as one. That's called summation. So, come back. If you have a single contraction, if you have a stimulation, which gives you a single contraction and relaxation, we call that a twitch. Obviously, you need a latency and so on and so forth. summation. This is where you add one contraction to another contraction. Again, you will get a greater force only because the amount of calcium that's still remaining in the sarcoplasm tends to get higher. If there's more calcium, there's more activation of the cross-bridge cycling, there's more activation of the actin, therefore the myosin can attach, and therefore you're going to get this increased contraction, and that's called summation. Okay? So, more action potentials, depending upon where they are, will give you a greater contraction. Again, single twitch, temporal just means in time. I gave one now, and I gave one there and one there. Or I gave one here, which I'll look at in the next slide. So if you look at this one now, again, I give a stimulation to the muscle, down the motor neuron, I get a twitch. I give another one, about, what is this, 100, about 40 milliseconds later, I get another contraction. But what's now going to happen is, I'm going to make the action potentials closer to each other. Notice what's going to happen. So I come here now, okay? I'm decreasing the time between the action potentials. So I'm adding this one to this one, okay? I keep decreasing the time. I keep adding the one to the other, yes? In other words, each time I give an action potential close to the previous one, I'm getting more and more calcium building up inside the sarcoplasm and inside the cytoplasm of the muscle. Another way of saying that is the sarcoplasmic reticulum is not working fast enough to pump the calcium back in and allow relaxation. Okay? So, again, increased force due to increased numbers of action potentials. The fancy way of saying that is just increase the frequency of action potentials. Okay? Again, if you do this on a single motor unit, I'll come to what motor unit means in a minute, then you will add this thing up as you go up. Okay? Why? Because the calcium cannot be pumped fast enough back into the sarcoplasmic reticulum, meaning it's there, meaning the actin binding site will be revealed, and you will continuously get cross bridges. And therefore, you will get increased muscle contraction or force. Are we okay so far? Good. Take this one step further now. Okay? Now, there are 4 diagrams here. You don't need to know all 4. I'm just showing you how this pattern can change. So in the first diagram, I've got a small twitch and a slightly bigger and a slightly bigger and so on and so forth. But each time, the action potential is a little bit further away, and therefore, the muscle can relax. So I have a relaxation here, and another one there, and another one there, and so on and so forth. Now, but it's going up slowly, so it's called a trep. Can you see the word T-R-E-P-P-E? It's a fancy word for stairs. Okay? All right. So now look at the same thing, but this time I'm adding. I make it close enough so that I don't get a total relaxation. So I add this one to the residual calcium of this one. I add this one to the residual calcium of this one. And it keeps going up again like a stair. Look at this one. Now I make the action potential so close, or the stimulation so close, that they add until I get to the maximum. We call that a tetanus, a maximum contraction. Again, look at the same thing, but now the action potentials are coming very close to each other, so that there is no relaxation. So now I have a straight line. Okay? This is called an unfused, an incomplete or an unfused tetanus. This is called a complete tetanus. The muscle never relaxes. Okay? Because all that happens is the calcium just keeps remaining in the cytoplasm. Okay? Good. So, you can get to a maximum contraction with a incomplete or a fused tetanus. So the word tetanus just means, in physiology, the word tetanus means a muscle is fully contracted. That's all it means. It doesn't mean you're going to die. It doesn't mean the muscle will stop. It just means it's fully contracted. Okay? Now, those of you who go to the gym and your gym trainer says, this last 1 minute I want you to run as fast as you possibly can. Are you with me?
Yes.
In that last 1 minute, you're just getting the maximum possible contractions in your muscle. That's all that's happening. And then you stop and everything's fine. That's what in physiology tetanus means, a complete contraction. Okay. So, again, you can have an unfused version where the action potentials are reasonably close together, but there's a little bit of a relaxation. Slide C, or diagram C in the previous slide. A fused contraction is diagram D, where there is no relaxation whatsoever. This generally happens at very, very high frequencies of stimulation. Okay? Again, 80 hertz. The word hertz just gives you frequency, number of action potentials per second or minute or whatever. Again, it occurs when frequency of stimulation is so high that the calcium cannot be pumped fast enough into the sarcoplasmic reticulum. The force that's generated can be about 5 times greater than a normal single twitch. I put down at the bottom, fused tetanus is not a pathological condition in physiology. But I put the second bullet point at the bottom. There is this toxin called tetanus toxin by a bacteria called Clostridium tetani that makes a muscle to contract to its maximum. And therefore, it's called the patient will have tetany or locked jaw, or the larynx doesn't work properly, and so on and so forth. Now that's a pathological condition. Okay? So, which now brings me to motor unit. So if you take a motor neuron and you stimulate it, the muscle will contract. If you stimulate it fast enough, the contractions will add up to the point of a fused, complete tetanus or a fused tetanus. Large muscles in our body, like quadriceps and biceps and so on and so forth, are made up of bundles of muscle fibers. And those bundles of muscle fibers have individual nerves that kind of innervate them. So, all right, a single motor neuron or motor nerve to the fibers that it goes to in the muscle will be called a motor unit. All right? Again, I mentioned to you in that little table when I gave you in the second lecture, in the summary, I said you can have motor units that are small. In other words, the neuron is going to few fibers. Or they can go to larger units, large fibers. Look at the bottom of the page, the innervation ratio. A motor neuron can go to maybe 10 fibers. Those would be the small ones. And that's generally for fine control. For people who play the guitar, the piano, or the violin, or, for those of you who will become excellent surgeons, that's what you're doing. On the other hand, high ratio, where you have one motor neuron going through 1,000 muscle fibers. Why would you want that? You would want that because you want to run or jump. Okay? All right? So those are coarse or gross movements. So again, small motor units. Okay, let me just get a pointer here quickly. Small motor units. All right? Again, for precise control, how are they stimulated? By low threshold. Remember my two little diagrams with the motor neuron coming here, and then you have low frequency stimulation, fewer acetylcholine vesicles. High frequency stimulation, you have a lot of acetylcholine vesicles. Again, they are fatigue resistant. They are fatigue resistant. In other words, they carry out oxidative phosphorylation for their ATP. They're required for fine control and so on and so forth. On the other hand, large motor units, okay? Again, less precision. You require a lot of stimulation for them to kind of contract. They can become easily fatigued, and they are required for large forces like jumping and running, like the quadriceps. Okay? Now, when a motor neuron is stimulated, all of the fibers that it goes to will contract. Okay? It's not like some of them will say, no, I'm going to relax this one out. No, it doesn't work like that. Okay? Now, so, look at that in another way. There's the muscle fibers. I have a yellow motor neuron going to 1, 2, 3, 4 of them, the red ones. I have a black motor neuron going to 3 of them. If you stimulate B, let's do B. Yes, let's do B. If you stimulate B, you will come up here, you'll get a force, and then you stimulate B again somewhere here. It's already relaxed, it goes down, and then you get another B. Are we okay? If you stimulate A, all right, there's A. No, there's A at the bottom. You stimulate A, all right, A is the solid line, there's A, okay, and I go somewhere here. I stimulate A again, but then I give another one close by to A, it adds. It summates. So I've got 2 motor units, okay, that have individual innervations on them, and I can kind of regulate the frequency of stimulations in each one of them. Okay, again, large muscle groups like quadriceps and biceps will have many motor units. In the muscle, you will have the slow ones, and you will also have, what is the correct name? The small motor units and the large motor units in that muscle. Okay? So, depending upon how the brain now decides to stimulate that muscle. Now, why am I using the brain? I came into this room at 1 o'clock. It is now 1:40. I've been standing all that while. Yes?
Yes.
Does that mean that only one group of muscle fibers are keeping me standing in that 40 minutes? No, I'd be tired. So what is my brain doing? My brain started off by stimulating the small motor units to allow me to stand. Then it switched. It made it a little bit faster. Okay? What were the small ones doing when the brain increased the frequency? What were the small motor units doing when the brain increased the frequency to the big motor units? What are the small ones doing?
Relaxing.
They are relaxing. Okay, they are relaxing. And then it will come back to the small ones. You understand what I'm getting at?
Yes.
Okay? And that's what I mean by spatial control. So the central nervous system, by regulating the frequency, will say, okay, I want this group, and that group to contract. Okay? Then I increase the frequency, I want that one, and that one to contract. All right? Then you come back to the small ones. And that's what I mean by the CNS will regulate this. So that not one group of fibers will get tired just doing whatever it is you're trying to do. Okay? So the number of motor units active at any one time determines the total force in the muscle. As more motor units are brought into the contraction, obviously you're going to have summation, okay, of the motor units, and this is what we call spatial summation. If you look at this in a slightly different way, there's it. So ventral, you have purple, blue, and red. They go into these fibers. Are we okay?
Yes.
You notice there are different numbers to each one of them. Obviously, you'll have a different amount of contraction for each one of them, okay, or total force. So if you look at that like this now, all right, So you stimulate, there's the muscle, and each little circle is like the motor units that are being stimulated. None are stimulated, none are stimulated. Why? There's insufficient voltage to stimulate them. Okay, fine. So notice no force. Now, here two are getting stimulated, I get a little bit of force. Yes? Again, if you look at the action potential, you're just about at the threshold. And those would be the small motor units. I increase the stimulation frequency. Now I come to this one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm getting more force. Okay? Again, there's it here. All right? And I keep stimulating, and there we go. And now I've added on the force of all of the muscle fibers in the motor units, and now I've reached the maximum. Okay? And that's what this is telling you. Again, the stimulation of more and more motor units increases the force because more parts of the muscle start coming into the contraction. Okay? So that's another way of looking at this thing. Okay. So motor unit recruitment. How does the brain go about saying, okay, I want you, you, and you to contract, but then I want you and you to contract in the next cycle? So each motor unit will contract in an all or none. If the nerve is stimulated, the fibers that it's going to will contract. Secondly, muscles will create a graded contraction, as I showed you in the previous slide. It will start off low and then it will become maximum. Again, how do you create this strong contraction at the end of the previous slide? You change the number of motor units you're using at any one time, or you change the number of motor units. A type of motor unit would be the small one, the intermediate one, or the big one. The number of motor units is how many you are using in each contraction cycle. How do you do that? How does the central nervous system do that? By varying the stimuli or the frequency of stimulation. Okay? So low frequency stimulation will be like around 8, 10 hertz or action potentials per minute, all right? And those will generally trigger the slow twitch fibers, the fatigue resistant ones, the ones that are using oxidative phosphorylation to generate their energy. On the other hand, as the stimulus increases, the number of action potentials are increasing, you now start stimulating the fatigue resistant, the fast twitch oxidative, the halfway in between ones. And lastly, at very high stimulation, you now stimulate the glycolytic, the ones that get tired very quickly. Okay? So if you ask me to jump like a rabbit to the door, I'll not make it. Okay? So the CNS avoids muscle fatigue by varying the contraction or the stimulation, and then the contraction of the motor units. We call that an asynchronous recruitment. So you go from small to large to intermediate, back to small to large, that kind of thing. In other words, you don't go one at a time all the way up. What are the motor units that are not stimulated doing? They are relaxing. Asynchronous recruitment prevents fatigue in submaximal contractions. If you look at these motor units now, and we're looking at these muscles in the legs, the quadriceps and so on, this one you notice is more red. If you look at the histology, it's red because it has more myoglobin, it has more capillaries in a blood supply, it has more mitochondria to generate ATP by oxidative phosphorylation. On the other hand, the fast twitch glycolytic ones, like the ones in the eye, okay, look, they have a large diameter, they are white because they have a lot of glycogen in there, so that the glycogen can be converted to glucose, the glucose can be used for glycolysis, okay? So, again, you have slow twitch, which are oxidative phosphorylation, and then you have these ones, the fast twitch, which get tired very quickly. And then you've got one in-between kind of thing. I didn't give you a diagram for the in-between one. So I extended my 2 little columns from lecture 2 to 3 columns now. And if you follow this thing, it says slow twitch oxidative. The red ones, the ones with the mitochondria, the ones with the decent blood supply, the ones with the lots of mitochondria. Okay? They tend to contract very slowly. Those would be postural muscles. The diameter tends to be small, but you have more myofibrils inside the muscle fiber. Contraction duration, long time, we can stand for quite a while. ATPase activity on the SR is moderate. In other words, it doesn't pump the calcium back into the sarcoplasmic reticulum very quickly, so you can maintain a contraction. The myosin ATPase activity, the one that's pulling the myosin head away from the actin, tends to be slow. All right? Again, where is this used? It's used in posture. Metabolism, oxidative, in other words, cytochromes and so on and so forth to generate ATP. Capillary density very high, mitochondria, lots of them, and generally they look dark. In my second lecture one, I looked at this one. Okay? And again, this one generates its energy very, very quickly. In other words, its speed of generation is very quick. The myosin ATPase is very fast in this one. Okay? Again, the diameter is very big in this one. Contraction duration obviously is very short now. You're going to get tired very quickly. Again, easily fatigued, only used in jumping, quick, fine, when you're running and that kind of thing. Glycolytic, anaerobic. So all that's happening is you're going from glucose to pyruvate and then lactate. Capillary density very low. You don't need the blood supply. And then you have few mitochondria and you don't tend to have a lot of myoglobin. And the one in between is like halfway between them. How does this relate to you as a doctor? If the motor unit is in any way destroyed, and that happens in polio, which is a viral infection of the spinal column, the motor units, the neurons will be killed off through the infection. You lose the motor units, and that results in an inability to contract the muscle. Again, summation. Why will a muscle, say in myasthenia gravis, not contract? Because you do not have sufficient summation of EPPs to give you an action potential, and therefore summation becomes a bit of a problem. Temporal summation, EMG, I'll talk about, we'll do this lab next week and I'll send you an email, but EMG, increased firing of a muscle, will tell you whether or not the problem is in the muscle or in the nerve, and I'll speak about that later. Tetanus I spoke about earlier on. Muscle tetany is a normal physiological function, but again you can have this pathological infection by this bacteria. And then the size principle, low frequency stimulation will give you the small forces, the fatigue resistant fibers will be stimulated. High frequency stimulation will give you the glycolytic fibers, and a lot of force is created. Let's leave the last slide for the next lecture, and that's enough.